Hi everyone. So by now, hopefully you guys are getting comfortable with the format of the class, and I hope you have already had the chance to not only take the practice quiz, but also the quiz on biomolecules and protein structure and function. Today's lecture will be moving away from the chicken and back to the egg, aka DNA. So today I'm hoping to talk about the history of DNA discovery and how we know that DNA is the molecule responsible for carrying genetic information. Also, how the DNA structure has been discovered and how the structure or the knowledge of the structure of the DNA influenced our understanding of the DNA's function. So let's start back in 1930s England, where Fred Griffith was working with pathogenic bacteria that cause pneumonia, and discovered that there's a mysterious transformative power that dead bacteria were able to have. But before we talk about his experimental results, let's talk about how he set up the experiment. He had two different strains of pathogenic bacteria. One strain, the virulent strain, was able to kill the mice when injected into them, and the other strain, the non-virulent strain, wasn't able to do so. The reason for this difference was that the virulent strain was able to produce a capsule that surrounded the bacteria and thus made it much more resistant to the immune system, to the mouse's immune system, while the non-encapsulated strain, the blue strain, here did not have the capsule and was easily cleared by the immune system of the mouse. Now, Griffith was also able to observe that if heat killed, the encapsulated strain also lost their ability to kill the mouse. The mouse lived when heat-killed encapsulated strains were injected into them, simply because the bacteria were dead and thus could not reproduce and overtake the mouse's body. However, he made a very important observation. When he mixed the non-encapsulated blue strain with the heat-killed virulent pink strain and injected this mixture into the mouse died, showing that somehow mixing dead bacteria was able to give the live non-pathogenic bacteria the ability to kill, the ability to have the phenotype that only encapsulated strain was having before originally. Now, the other observation that he made was that not only did the phenotype changed for the living non-virulent bacteria, the R strain, they were now able to kill the mice, but that the cells, that the bacteria that he then isolated out of these dead mice had changed dramatically as well. They no longer looked like the yellow R strain. So in this picture, this is yellow. In the previous diagram, it was blue. They no longer looked like the non-encapsulated, non-virulent R strain. They now looked like the virulent S strain. And the only way the S strain made it into the whole mixture was after being completely killed off. He made a lot of tests to make sure that the injections that he gave the mouse did not actually contain a few of the red S strain. In the previous picture, it's red as well, and was able to show that indeed somehow whatever the dead husks of the S strain contained was able to change the living R bacteria. So this was a surprise number two, that the live bacteria recovered from the dead mouse changed. And it was a permanent change, because when these now pathogenic S strain bacteria, former R's, were used to infect other mice, the mice were dead. So the question became, well, what was it inside the cellular husks of the red strain that was heat killed before it was injected into the mouse? What molecules, what factors inside this mixture were able to transform the non-virulent R strain into a virulent one. And these experiments the experiments to answer that question were then performed in the 1940s in the United States by Avery, McLeod, and McCarty. They were good biochemists and very good at purifying different biomolecules out of cells. So they grew up vast amounts of the S strain cells. The heat killed them, just like Griffith did. And then they fractionated their soup of the dead S strains using various approaches: centrifugation, precipitation, columns, whatever was available for them at the time. And then they tested the different fractions that contained different molecules by mixing only that fraction with the naive R strain and injecting them into the mice and seeing whether that fraction of the S strain was able to transform the non-virulent strain into something that would kill a mouse. And they observed that only when the DNA fraction was mixed together with a non-virulent R strain bacteria, as you can see here, when this cocktail was injected into the mouse, only then the mouse died, and only then did the R strain cells, the non-virulent R strain cells, changed into the S strain. Other types of biomolecules did not have that power. And the conclusion of the Avery, McLeod, and McCarty experiments was that DNA was able to carry heritable information. But there was still a nagging question. A lot of scientists discounted DNA as the molecule to store and inherit the genetic information, and were thinking that proteins were much more likely to do it, simply because proteins were more complicated. Their reasoning was that how can something that's so simple, that is made out of 4 different nucleotides, contain enough information for making proteins that are made out of 20 different amino acids? Again, how could something that only contains AGCT control the making of different lipids or the different carbohydrates that the living cells contain? And this question was in understanding the makeup of DNA was something that another United States scientist, Irvin Chargaff, was very interested in. He was also working in the 40s and the early 50s, and he discovered through his sets of very careful measuring experiments 4 different rules of nucleotide base composition in all living organisms. He compared lots of different species, from plants, bacteria to humans, and realized that first, that the base composition of DNA, the percentage of A, G, C's, and T's, were different between different species, which is not surprising because our genomes are different. That's what makes us different. And because our genes are different, slightly different at certain levels, we will have different amounts of A's and T's and G's and C's incorporated into our DNA. Some organisms have a GC-rich genome, some organisms have an AT-rich genome. Also, he was able to observe that when DNA samples were isolated from different tissues but from the same animal, the base composition was the same. So if you take, and that's again not surprising, every living cell in your body contains exactly the same DNA. DNA is not changed unless there is certain mutational errors during DNA replication. Every cell in your body contains the same amount of DNA. Which part of the DNA the cell reads is a different question. That's what makes the different cells in our body look different and have different functions, because the mRNA produced from the DNA is different. But the base, the DNA content of the nucleus is exactly the same. He also measured whether the DNA at the base composition changes with age or nutritional state or the environment of the organisms, and found that there were no changes. So DNA the base composition of the DNA molecule, of all the DNA molecules in your body, are very, very stable, which is great because that's what we need DNA to do. It needs to be stable, non-reactive, conservative, and unlikely to change. Because frequently the change does not bring anything good with it. And then the final rule, rule number 4 of the Chargaff rules, said that in all forms of DNA, and he did a lot of work, regardless of species, there was always the same amount of A's as there were T's, and G's as there were C's. Now again, that's not a surprising finding at all, because we know that inside DNA, an adenosine will always only pair with a thymidine, and a guanine will always pair with a cytosine. And I have to say that there is a lot of information that was gained just simply by carefully counting A, G, C's, and T's in different organisms and thinking about it. Now, the MacLeod work, the Griffith work, and the work of Chargaff all boiled down to a realization in the 40s and then 50s that DNA had a very important secret inside of it. And by understanding how the DNA molecule actually looks like, it might be possible to understand how it could act as a transfer of hereditary information. So in 1952, Rosalind Franklin was able to capture image 51, which is this photograph here. And I think this is the kind of photograph that every molecular biologist needs to have on the wall of their house. It's both very modern and very classic. And so the image 51 that Rosalind Franklin was able to capture showed that the DNA molecule, as it was diffracting the X-rays, was very symmetrical. Now, when Watson and Crick, the famous discoverers of the DNA structure, became aware of the image, they started proposing different potential structures, and they were wrong for the first few. In fact, they were banned from working with DNA due to the slightly illegal ways that they obtained image 51 from Rosalind. But then Watson and Crick also became aware of Chargaff's rules. They met accidentally, so Chargaff was visiting England. He met with them because they were curious about talking to the DNA, and he described what he had found. And the realization came that Chargaff's rule number 4, that stated that there are always as many A's as there are T's, and there's many G's as there are C's inside a DNA molecule, suggested to them that there had to be a base pair-like arrangement. And that is how the structure of DNA was discovered. So what I just told you is only a brief summary of what the DNA episode 1, The Secret of Life, covers in about an hour-long PBS documentary you can see on YouTube. These are the photos of the 3 guys that got the Nobel Prize, the Watson, Crick, and Wilkins. This is an image of Rosalind Franklin, who died very young, and of whom not very many images survived, simply because she was 37 when she died from ovarian cancer. And also Chargaff. So these 3 were recognized for discovering the structure of DNA, and neither Franklin nor Chargaff were recognized for their contribution to the structure. And here I'm going to take a little break.